an injury. So while you can do that visually, more recently, approaches have also been developed to do more with that and to learn more out of these signal intensities, but also to analyze them more accurately and automatically. So today we will discuss 3 papers of them. One I will just briefly mention. It's for you to read the details. I'm not going to pretend that I'm an expert in machine learning. I'm not at all. I'm just a novice there and trying to learn as much as I can. So what this group did, and it's a Chinese group, and our Chinese journal club members, they typically do not watch that on live. They watch the recorded version, so I already welcome them. And want to also specifically thank this group for this work. So what these people did is they looked at T1 maps in patients with dilated cardiomyopathy and compared them to T1 maps of a group of healthy people. So specifically, they had 50 patients with dilated cardiomyopathy and 24 healthy controls. And then they performed a standard protocol that included T1 mapping, and then they looked for certain characteristics in the texture, so in the structure, the apparent structure of the T1 times. And they identified 4 main categories. That was model structure transformation statistics. And then they had certain features they used to train a learning algorithm. And these features were based on texture alone, but then also they analyzed the histogram, also with certain features, standard deviation, minimum, max, and then the percentiles, and then mode, skewness, and kurtosis. So they used a home-made software MATLAB tool, and so this is what the flow diagram, what they basically did. And then they looked at the end with, of course, in a fairly small sample size, what the accuracy. And what they found is that when they used this model and they transferred to the histogram, that indeed there was a difference between dilated cardiomyopathy and healthy controls. But as you can see, there's also quite a bit of an overlap. But interestingly, when they combined all the different features, then they came to an accuracy, an achievable accuracy of about 85%. That's not stellar, but given the fairly small amount of identifiers and the small sample of that they could use for teaching the algorithm, I thought that was pretty interesting. So with that, I would like to hand over. I will stop sharing my screen. I will hand over to Reza and his team to present their 2 papers, and one was published in the Journal for Cardiovascular and Magnetic Resonance, and the other one in the Journal of the American Heart Association. And so I will hand it over to them, and the first one maybe, if possible, by Dr. Fahmy on automated analysis of cardiovascular MR, native T1 mapping using fully convolutional neural networks. So I hope that works. Can you take over and share your screen? Reza? I think you have to unmute yourself, or no, I think you're already unmuted, are you? I can't hear you. Can you text in the chat whether there is an issue on your end?
So what do we share?
Is that you?
This is Dr. Neisius and Dr. El-Rewaidy. We are supposed to present the second one.
Okay, great. Yeah, so no, then I think this is your paper, isn't it? The automatic analysis of cardiovascular MR with fully convolutional networks?
It's not. It's they are still trying to get established.
Okay.
So
then let's start with the other one. Yeah. So that's great.
If you want, we can start with that while they're still
if you can just share your screen.
Okay.
So, and then walk us through a PDF.
Okay. So basically, this paper was published 2 months ago online at JACC Cardiovascular Imaging. As already introduced, we addressed a clinical problem, the differentiation between hypertensive heart disease and hypertrophic cardiomyopathy with radiomic analysis of myocardial native T1 images. Just to summarize, basically, there is a clinical need for improved differentiation between hypertensive heart disease and hypertrophic cardiomyopathy, which is based on the fact that in roughly 25% of cases, hypertrophic cardiomyopathy patients have actual hypertensive heart disease, and the fact that only 60% of hypertrophic cardiomyopathy patients are actually sarcomeric positive. This can be established by imaging, and there are lots of features that are already investigated and dedicated to this problem. But we try to address pathophysiological or histological abnormality of the hypertrophic cardiomyopathy patients. Basically, their fibrosis pattern is quite different from what is established for hypertensive heart disease. Hypertensive heart disease is more a continuous interstitial fibrosis, whereas in hypertrophic cardiomyopathy, this is quite patchy, based on myocyte disarray and fibrotic patches on probably mechanically weak areas. And we thought we could actually identify these histological differences by measuring using native T1 maps. Native T1 maps, because these are quite well established as fibrosis assessment tools in both diseases, and the literature is quite wide on that field, so there seems to be a lot of evidence for that. And then apply radiomics and radiomics approach to the differentiation between these two diseases. Radiomics in general is motivated by the concept that biomedical imaging contains information reflecting on disease-specific processes, and which can be accessible by quantitative image analysis. This field is predominantly dominated by oncology imaging, and the literature goes back, I would say, 10, 15 years. And we are more or less one of the first ones that try to apply this technology or this post-processing approach to a cardiovascular problem. So what did we do? We had a quite big cohort of hypertrophic cardiomyopathy patients where we had set T1 images, 108 in total. We defined them by the current guidelines, which are basically clinically diagnostic tools plus imaging. Then we collected a few patients with hypertensive heart disease. We defined hypertensive heart disease as patients with increased wall thickness on cardiovascular magnetic resonance imaging in the context of hypertension and no other disease that could contribute, such as aortic stenosis, etc. And then to set this all into context, because we obviously don't know what normal values are, we investigated a control group which came through our referral system here with completely normal diagnostic scans and no cardiac history whatsoever. And then, sorry, I'll come back to that image later. Then we performed on 1.5 T-scanner native T1 maps with a slice interleave T1 mapping sequence called STONE. And we covered the whole heart from base to apex. And here we go further. And then we performed feature extraction. Basically, sorry, can we just move that? No, we can't. Based on 4 sets of feature texture descriptors. I'll come to that later. I'll jump over to a slide to explain how we first did the image pre-processing before we performed the feature extraction. Can we share over to the... wanted to show this one. Yeah. Can we magnify that? Basically, so you can see, no, that's, sorry, this one? Okay, we basically performed T1 mapping as shown here, then define the region of interest, APA in blue and endocardial contour in red, then split this region of interest open by starting at the inferior insertion point and created a stacked image and created a sort of rectangular image of the region of interest, and then from base to apex stacked these together into a map, which then on which we then applied the feature extraction. Here you can see that these maps look quite different, where the hypertrophic cardiomyopathy map is more grainy and more heterogeneous, whereas the hypertensive heart disease maps were quite homogeneous. Let's go back to the, how do I share my, okay. And if you do that, and then we applied the features, and we used 4 feature descriptor groups. So they are table 1, which are histogram features, run length matrix features, co-occurrence matrix features, and local binary patterns. And these were then quantified, and the results showed, and we used all these features, in total 152, to select a group of features that best identified hypertrophic cardiomyopathy patients, and we ended up with a total of 6 features, which had a diagnostic accuracy of of 86%. Here you can see how each of the features contributed, was sort of quantified for each group, and you can see that for most of these features, of the 6 features identified, the difference was quite significant. So we had to Bonferroni adjust because of multiple testing for the features, and with the exception of 1 feature, all were significant between both groups. And then just gonna, here you can see, if you do for the features, that these accuracies were quite good. This table needs a little bit further explanation because of the omics approach of, we had to sort of validate that our assessment was to some degree generalizable in the open field, and we therefore split our whole cohort into 2 groups. One was a feature selection cohort, which were contributed by 80%, and the other one was a test validation cohort, which was contributed by 20% of the original cohort. And the feature selection was performed only on the 80%, and then we used the sort of risk stratification process, which we sort of developed in that feature selection cohort and applied that to the test validation cohort, and got a drop of accuracy of roughly 6%, which is frequently seen in omics research. Basically, if you apply or define an omics marker, and if you try to replicate that in a separate cohort, you usually lose a little bit of test accuracy. And then, interestingly, these test accuracies have been much better than for the native T1 assessment by itself, which is illustrated in figure 3. Here you can see that on the left side panel, the global native T1 for hypertensive heart disease in comparison to hypertrophic cardiomyopathy patients has a significant overlap. And if you use our features summarized by an index, you can see that the difference between the cohorts is much higher and enlarged, which gives us an argument that for certain disease classification, radiomics approaches help to differentiate between diseases beyond global native T1 mapping itself. And that's, so in summary, we had 2 main findings. In this proof of concept studies, we could show that tissue analysis is feasible for native myocardial T1 mapping, and that per se, it helps to discriminate between hypertensive heart disease and hypertrophic cardiomyopathy, and B, that these features can provide incremental value over global native T1 mapping, although admittedly this is only for hypertrophic cardiomyopathy to hypertensive heart disease comparison. That has to be reinvestigated for basically every disease cohort to make certain that this can be applied. Thank you. I hope that was.
Thank you. Yeah, thank you very much. That was it's very interesting, and congratulations, and thanks for this important work. So in a nutshell, so for people who are not that familiar with machine learning, so can you briefly describe, because apparently what you're doing is you're identifying certain patterns or characteristics in the T1 maps that further improve the accuracy of the map to identify or to discriminate between the two diseases. Now, these so-called features, did you use, and I may have missed that, did you select features, or did you use features that were provided by the machine learning system, or did you use what's called unsupervised learning, that you left it all to the system to find out which features would best separate that?
Okay, thanks for the question. I didn't address that because it's quite extensive explanation required. But basically, we applied features that we well predefined. We basically the features by itself are the way we use them are mathematical equations, and you then apply them to the region of interest. I can show just to illustrate.
But in the interest of time, we will have to move on soon to the other paper. But did you yourself define these features, or were they somehow pre-specified in the algorithm?
They were pre-specified. They are frequently everywhere in the literature, basically oncology literature, as mentioned before.
Okay. So you use standard features that were already part of the system.
Yes.
Okay. Yeah. Thank you very much. So thank you so much. And now I would hand over. So if you stop sharing, please. Hossam, then we would I would ask Reza and his team to share their screen for the other paper. Oh, it's the same. That's great. You're sharing the system there. That's good.
Yeah.
Hi. So maybe you could just briefly also introduce yourself while you're getting ready.
Yeah.
And we have only 10 minutes left, so we'll have to speed up a bit. Maybe while we're waiting, I know that we have a couple of machine learning specialists on the call. Any questions to the previous paper? Just speak up, unmute yourself and speak up, or raise your hand in the chat, or just type it into the chat, then I can ask the question for you. Okay, if that's not the case, then we can move on to the next paper published in JCMR. The first author is Ahmed Fahmy, and it's again from Reza Nezafat's team, and it's entitled Automatic Analysis, Automated Analysis of CMR, Native T1 Maps Using Fully Convolutional Neural Networks. Take it from here.
Yeah, hello. My name is Ahmed Fahmy, the first author of this paper. So I will try to be brief, and then if anyone have any question, he can just ask, and I will elaborate more. Here, okay, before I discuss this paper, I just want to mention that we in this work we relied on the reliability and the robustness that was shown in many papers before on using the deep learning for segmenting the myocardium. So we employed this robustness of segmenting the myocardium in T1 mapping pipeline. So it's basically segmentation of the myocardium, and then we added some functionality to allow reliable or fully automated mapping of the myocardium T1. So at the core of this work is a fully convolutional neural network to segment the myocardium, the T1 weighted images. For this segmentation, we used that set for 210 patients to just train the network and test it for the segmentation. And after that, we used an independent dataset for 455 patients to test the calculated T1 values using this pipeline. We did the analysis on per patient and per slice basis, and we also selected a subset of the dataset, and it was manually analyzed by a second reader so that we can compare the interclass correlation between the automated method and the two readers, with each other. So the summary of the results here, the first interesting was the very fast segmentation and computation of the T1 mapping. So it took only 0.3 seconds to segment one image. And we have a reliable, accurate segmentation of the myocardium. And for the T1 values calculated by the automated method and the manual analysis, we had very strong correlation between both of them in the per patient analysis, and a little bit lower correlation for the per slice. Also, the interclass correlation between the automated method and both readers were comparable to the interclass correlation between the two readers. So this is the summary. I will now go back to the methods. And I prefer to start from figure 1. Okay, so this is the pipeline of the analysis. So at the beginning, we have this T1 weighted image input to the neural network. We do segmentation. So we mark all the pixels belong to the myocardium. Everything else is 0. And then we do some testing for the resulting shape. We assume that the proper shape of the myocardium is this donut shape object, which is a regular circle or ellipsoid. It's not very elongated or one patch of object. It has to be of this regular shape of the myocardium. If not, and this happens in many slices, we do some refinement. And this refinement is based on taking the best segmentations that we have, proper segmentations that we have for this volume, and then try to kind of interpolate or fill in the missed parts of the myocardium, so that we reach a final segmentation that is a donut shape, annulus shape myocardium. So after making sure that this segmentation is correct, we do a polar transformation of the myocardium in the input image. So let me switch to one of the additional files. So in this polar transformation, we sample the myocardium from this location, arbitrary location, in a circular order. So we take all these pixels and stack them in a rectangular matrix. In our case, we use a dimension of the matrix of 20 by 360. So we have a ray that rotates by 1 degree, and then we sample all the intensities along this ray and stack it here to have this rectangular matrix. We do this for all input T1 weighted images. So in this case, all the images for a particular patient, for a particular slice, are inherently aligned or registered in this polar coordinate system. So after we do this polar transformation for all T1 weighted images for a given slice for this patient, we have for the image acquisition sequence that we use, we have 11 of these T1 weighted images. So after taking them, we do pixel-wise curve fitting for each pixel in this set of images, and then we obtain a T1 map for this slice, and then we do inverse polar transformation to get a very nice regular map. But this map is only for the myocardium. It's not a whole slice T1 map. So yeah, these are details of the each block in the pipeline. And this is the neural network that we use. It's based on the famous commonly used UNet architecture, where the input image is input to the network with a full size, and the output is a full size image, segmented image. Of course, we do resampling of the image to be 256 by 256, just normalization of the input size. Okay.
Just as a heads up, I'm so sorry, but we only have a few minutes left, so just skip some of the detail and the high level, so that we have some time for some questions maybe.
Okay, sure. So for the segmentation accuracy, as you can see here, this is the dissimilarity coefficient, which gives us a measure of the similarity between the manual and the automatic segmentation. As you can see here, we have very good segmentation accuracy, but as you can see, it's lower a little bit for this inversion times, where there is low contrast between the myocardium and the blood and the background. So we have here lower accuracy in segmentation, and also we have lower accuracy in the apex compared to the basal and mid cavity slices. I want also to show some of these results. This segmentation, as you can see here, it's very nice segmentation, even if the contrast between the myocardium and the blood is very low, we still can perform very well for segmenting these images. And this is the resulting maps. So we get map for the myocardium, and then we remove the boundary pixels to avoid the residual errors related to imperfect segmentation at the edges. So we get this region of interest within the myocardium. And this is the manually analyzed maps. And this is the region of interest placed by the reader. And this is a plot of the automatic T1 mapping values per patient and the manual analysis. And this is in the pair slice analysis. We have correlation coefficient of 0.8 and 0.7. And this is the bland Altman plot for these results. And finally, this is the ICC between the automatic and the expert or manual analysis, as between the automatic and the manual analysis of the by the second reader. And this is the ICC between the two readers. And as you can see here, the automatic segmentation is comparable to the variation in the per patient and per slice. That was based on 40 cases. 40 cases, and we did the manual analysis twice for the subset. Yeah.
Okay, thank you very much. So in the interest of time, we'll have to keep it short, but first of all, so the takeaway here is that an automated approach, including network-based deep learning, is maybe just as good as another reader, another experienced reader, so to say. Your sample size, that may be a question also to Reza, if you could chime in here, felt still on the low side. Do you think that these results could be even further improved by larger samples? Or don't you think it makes a big difference?
You're talking about the network, the training of the network, right?
Yeah, yeah.
So actually, okay, this is a good question. Actually, we have this 210 patients for the training, but for the training and testing, but we use only 63 cases for training, which is 30% of that set, which is smaller relative to what's usually done in this field. However, the initial network that we used here was pre-trained from our previous work on LGE, where we segmented the myocardium and the LGE in LGE images.
Okay.
So we started from a very good initial weights of the network. I don't think if we increase the number of cases, probably we need more diverse cases for other diseases, larger cohort of patients. However, other than that, for this particular sample, we don't think we can improve the results a bit.
Okay, yeah. Any other questions from the floor or any participating? I think.
Hi, so maybe a really quick question. Thank you so much for the nice presentation. My question was about the polar map. So, what is the reference point or 0 degree for the polar map? Do you use the insertion points, or do you always have a certain assumption to start your polar map?
We actually it's arbitrary. We start at 0 angle, like perpendicular to the x-axis, and we just do the rotation of the.
So then the assumption is that heart doesn't rotate, or there is no deformation in myocardium thickness during the inversion times.
Not the thickness. The assumption is there is no rotation between the different T1 weighted images, which is a good assumption, I think, because we always image at the end diastole, and patient usually the respiration motion does not affect the rotation of the myocardium.
Have you thought about some sort of joint segmentation and motion correction? There is paper also from the, so in the MICAI doing some sort of joint segmentation and motion estimation. So to do registration and motion, I think the polar map idea is to solve some sort of registration problem, right?
Yes, actually.
So how do you think machine learning or deep learning convolutional neural networks can be used to address that?
For the registration, if you're talking about using the neural network directly to register the images, I think it needs a lot of development, because the using, for example, the recurrent neural network to deal with sequence of images, training of recurrent neural networks is still complicated. So I don't think it's straightforward to apply neural network for registration. The state of the art now is to use it for segmentation. It performs very well. So at least at the current stage, I think segmentation and then use the segmentation-based registration is the optimal way of doing it. Doing it for registration probably needs a lot of future research.
Okay, thank you very much. So in summary, if you could also unshare your screen then, because I would just briefly highlight one. So in summary, definitely the machine learning for tissue characterization, specifically mapping, is an emerging field. It is important. At least I came to the conclusion that deep learning will infiltrate everything we're doing in CMR. And I just want to show two two papers. One paper I want to highlight that, and you can see it here, as a primer for those interested in machine learning or deep learning and in cardiovascular medicine. So here is a nice review paper published just I think a few weeks ago in the European Heart Journal that gives an idea of what deep learning is about, explains some of the most important keywords there, and explains also the basic structure of these approaches. So I highly recommend a read. It will be also available for download from the CMR Journal Club website. And then I also want to highlight a brief paper by Chandra, the editor-in-chief of JACC Imaging. It's basically an overview of what he thinks will be interested in CMR for the upcoming year. So that's a nice, also a nice view on how an imaging expert who's not necessarily only doing MR is seeing the field. And for next week, I want to already show two papers we will discuss there. They are also related to mapping. And one will be from multi-center side, looking at the role of T1 mapping in the diagnosis of heart transplants. So that's very important. We had mapping has been done, or tissue characterization since a long time. Now it was time for a multi-center study with histological validation that has been done here. So we will discuss that. And another paper by the group of Eike Nagel and Valentina Puntmann, looking at looking at the combined role of T1 and T2 mapping in cardio-oncology. So these will be the papers we will discuss in 2 weeks from now. And I hope to see many of you there. I want to thank cordially the presenters today, and also the discussants. I want to thank Reza for his specific suggestions for today's journal club, and then wish all of you a great day or a nice evening, depending on